Okay, morning guys. Um, let's pick up in Unit 4 where we left off last time, talking about microorganisms and disease. And we were talking about aroma flora and where we could find bacteria, where we could expect to find different concentrations of bacteria in the body. And I left off talking about started talking about the digestive tract. And we started with the buccal area, which of course is the mouth and the back of the teeth. The teeth and the back of the mouth. Um, and you can see here we've got lots of different species, 100, 200 different species, and we have a lot of numbers of bacteria here as well. 100,000 bacteria living on each tooth surface. That's not counting what live um, on your tongue, in the gingiva, on your cheeks, and so on and so forth. So there's a lot of bacteria that live in your mouth, and the reason why is because it provides a great environment for them. There's lots of nutrition there. You're constantly putting food in your mouth. It's nice and warm. It's got lots of moisture. So it's a perfect place for bacteria to grow. Now, as we move down the digestive tract, um, you're going to see a lot of variation between person to person of what bacteria actually live in their gut. Um, and the reason why that is because every one of us live a different life. We experience different things, we eat, we taste different things, and this will contribute to the bacteria that grow in your gut. So some of the things that'll affect the flora of your digestive tract is, for example, your age. The older you are, the more exposed you are to different foods, to different tastes, um, the type of diet you consume. I grew up in Canada. You guys didn't. I would assume there's going to be a difference in our bacterial flora or digestive flora because of that. The type and frequency of medications can have an effect. If you're taking an antibiotic for a long time, you're going to diminish some of those bacteria. Um, and then there's cultural and lifestyle conditions that can make a big difference too. Um, some people eat at home and they eat very healthy. Other people eat out all the time and are always ordering everything from Talabat. Um, it's going to make a difference in your digestive tract what bacteria are present there just because of that. Smokers will have different gut bacteria than non-smokers. People who are highly stressed or obese, they'll also have different bacteria based on their physical condition. And finally, the geographic location, like we talked about earlier, where people grew up, the type of food that's consumed in China, in the Middle East, in Africa, in North America, even in South America, it's going to be quite different. And those variations in diet are going to have an effect on what bacteria live in your gut. So it's very, very interesting, this topic to study, because your lifestyle and where you're from has a big impact, a big role to play. All right, so if we have a quicker or a closer look at the digestive tract, if we obviously we leave the mouth, we go down through the esophagus, and the first place we end up with is the stomach. Our stomach is a bit of a defense for us because the gastric juice present, the enzymes present, it makes it a very hostile environment for most bacteria. It will kill and disrupt the growth of most bacteria because of that very low pH. Now, some bacteria, those acidophiles, they have adapted to live in those high pH environments. And one species in particular is Helicobacter pylori. There's a very interesting story about Helicobacter pylori. As far back or as recent, I guess you should say, is the 1970s and 1980s, there was some controversy about stomach ulcers. Some physicians thought that they were caused by stress, and some thought they were caused by bacteria. So two doctors came together and started working on this project, the stomach ulcer project, and the two guys were Robin Warren and Barry Marshall. And Barry Marshall they were both of the idea that it was bacteria that caused stomach ulcers. And in particular, it was this one right here, H. pylori. So they got together, and the work that they did, it suggested that H. pylori was the cause of ulcers. Barry Marshall wasn't having much luck in the lab testing this with mice, so he decided to recruit a human being for his study, and the only one that he could recruit was himself. So what he did was he drank a culture of H. pylori. As he hypothesized, the bacteria set up in his stomach. It caused all the serial stages of ulcers. Eventually, he developed an ulcer. He biopsied himself, cultured the bacteria from the ulcer, and then he treated himself with antibiotics, and the ulcer was cured. So he proved, using himself, that H. pylori caused stomach ulcers, and it can be treated with antibiotics. And that happened in 1981. And in 2005, they shared a Nobel Prize in medicine for discovering the cause and the cure of stomach ulcers. Don't try that yourself, okay? If you think a bacteria is having a negative effect on your digestive system, don't culture it and drink it. Save it for the professionals. Now, moving past the stomach into the small intestine, of course, you get the duodenum, the jejunum, and the ileum. We won't see any bacteria, very few bacteria in the first part, the duodenum, but we will see some in the jejunum and the ileum. Staphylococcus, Lactobacillus, and Streptobacillus, these are all species that we commonly find in the digestive system. If we pass through the small intestine and get into the large intestine, the colon, this is where we're going to find the highest numbers of not only pathogens, but the highest number of bacteria and the greatest number of species as well. You're going to find a tremendous amount of Escherichia coli or E. coli in your large intestine. Also going to find over 600 different species that live in the colon. Almost all of these will be anaerobes, so they'll be living anaerobically, and they'll live off the digested waste in the bowel. They'll ferment that, use that for energy. And for us, some of their excretions, for example, E. coli, will help us produce stuff like vitamin K. So we get some, they get some use, they get a place to live and food, and we get excretions like vitamin K, which is good for us. So this is one of those mutualistic relationships that we were talking about a few slides back. So we consider this a positive relationship. Now, there's so much bacteria in your colon, just to give you an idea, that 50% of your fecal mass, if you were to weigh it, 50% of that number would be bacteria. So it's a massive, massive amount of bacteria that live in your lower intestine, in your colon. Moving on to the urogenital system. So this is going to be the system that makes up your urinary system and your reproductive tract. So now we're talking about, if we look at the kidneys first, the urinary system, the kidneys, the ureters, the urethra, the bladder, all of these should be sterile. We shouldn't see any life or bacteria living in those. However, the opening of the urethra, so that's where your urinary system exits the body. We will find microbes back there. We'll find bacteria. We can find fungi. We can find viruses. But that's to be expected. The constant flushing of the urinary tract, so you're constantly producing urine, it's moving through the urinary tract. This will prevent a buildup of cells, a buildup of pathogens, and that will protect us from infection. So if you're using the bathroom consistently and you're not dehydrated, that's going to help you prevent a urinary tract infection. A UTI, or urinary tract infections, these occur when normal flora, the bacteria that we expect to see at the mouth or near the mouth of the urethra, causes a blockage. And that blockage will give space and it'll allow bacteria to grow there, and that can give us the infection and then inflammation that we would associate with urinary tract infection. Okay, so that's our normal flora, or a little bit of an overview of what you can expect to see on a normal flora. So we'll go to the second half of this unit now, and we're looking more specifically now at microorganisms and diseases. So one question I'll have for you now, and I'll go on to answer this in the slides, but what's the difference between an infectious disease and a non-infectious disease? So this is a really good exam question. If you're watching this video, you might want to put a star next to this, so make sure you understand these concepts. Okay, so how are these two ideas different? Well, an infectious disease is caused by an organism that can be transmitted from person to person. Okay, um, you'll often hear this called a communicable disease. Um, this can occur through ingestion, through bites, through coughs, etc. And I guess the really important or really relevant example of an infectious disease right now is COVID-19. Okay, so that's spread from person to person by an infectious agent, the SARS-CoV-2 virus. A non-infectious disease is caused by something inside of you that's not communicable. It's not going to be transferred to person. It can be caused by your genetics, it can be caused by something in the environment, or it can be caused by lifestyle. But it's non-communicable, so it can't be passed from person to person. It can be caused by stress, genetics, allergies, or nutritional or metabolic issues. So an example of a non-infectious disease would be cancer, for example. You cannot catch cancer from somebody else. It's a result of genetics or something that happened in the environment or your lifestyle. Another example would be hypertension. So hypertension, of course, is high blood pressure, which can lead to a host of other complications. So those are examples of non-infectious or non-communicable diseases. Now, when we're talking about communicable diseases, of course, we need to talk about an infectious agent. And an infectious agent can be a virus, it can be a bacteria, it can be a protozoan, it can be fungi, it can be worms. We're generally going to be more concerned with virus, bacteria, protozoans, and fungi. The process of infection, how these cause disease, are going to occur in different stages. First, you need to be exposed to the infectious particles. This is the part right here that we are all trying to prevent right now when we're talking about our COVID-19 example. Okay, we want to reduce our chances of ever being exposed to this virus. That's why we are washing our hands. That's why we are staying inside. That's why we're canceling flights and closing the grocery store, closing the malls and the cinemas, and so on and so forth. We want to stop this break in the chain. We want to break this chain, this part of the infection, so that we can give this virus enough time to hopefully find a vaccine so we can go back to a normal way of life. But the first step is going to be an exposure to the infectious material. The second step is when those infectious agents establish a focus of infection or colonize some surface and overcome the host defenses. At this point now, you've been infected by this agent and it's going to go on and cause disease. If we're talking about, to be general, the surfaces where these can colonize can be skin, or it can be mucosal membranes. So it can happen on the skin, the gastrointestinal tract, respiratory tract, urogenital tract. All these areas are going to be susceptible to a possible infection.